All right, great. So, hi everyone. My name is Tomiko Oskotsky, and I'm a research scientist in the Sirota Lab of the Baker Computational Health Sciences Institute at UCSF. And I am very pleased to be here with Alennie Roldan. She is formerly a bioinformatics programmer in the Sirota Lab and is currently a medical student at the Burrell College of Osteopathic Medicine. And it is our privilege to share with you our data sharing and crowdsourcing approaches to advance preterm birth research. We're thrilled to talk with you about our Preterm Birth DREAM Challenge that leverages publicly available microbiome data from multiple sources, including from our Marchadigm-sponsored data repository. First, some background information. As you may know, preterm birth is when delivery of a baby takes place before 37 weeks of gestation, and it's a significant cause of infant morbidity and mortality. It happens in approximately 10% of births worldwide and can result in a variety of adverse outcomes, such as respiratory illnesses, blindness, and cerebral palsy, with associated complications resulting in nearly 1 million deaths per year. It's been found, it's a condition that can affect families as well. And it's been found that preterm birth may be associated with women who are on the younger or older end of the age spectrum, with smoking, and with twins, triplets, and other multiples. However, preterm birth is a condition that has been a challenge to understand, let alone to predict. The microbiome is a collection of all microbes, such as bacteria, fungi, viruses, and their genes that naturally live on our bodies and inside us. The microbiome may be mechanistically related to preterm birth. So here's a question. How can we predict those at higher risk of preterm birth based on the microbiome? There have been a lot of studies for preterm birth, but studies have been inconsistent in their findings. And due to their limited power and representation of populations, findings from these studies have not been generalizable. Combining data from multiple microbiome data sets would be a key step to having the results be generalizable to people all around the world, to people from different racial and ethnic groups, and more. However, combining data from microbiome studies is particularly challenging. Here's an example of a meta-analysis that was done by a very talented scientist who completed her postdoc in the Sirota Lab, Edith Kosti. She and her team found that bacterial vaginosis was associated with preterm birth. And also, they saw that there was higher bacterial diversity associated with preterm birth, particularly in the first trimester, as you can see in the graph here on the right. For this meta-analysis, Edith and her team used a mixed effects model where a lot went into adjusting the data by study and some information about the microbiome was lost. This theme is, I guess, repeated. The keynote speaker mentioned this as well, about losing some biological signal when you try to homogenize data across for technique issues. So for our preterm birth challenge, what our team wanted to do was to identify multiple vaginal microbiome data sets and combine them in a way that could preserve as much of the biological signal as possible. And that brings us to the Preterm Birth DREAM Challenge, and I will pass it over to my colleague Alennie, who will tell you more about the data.
Right, so as Tomiko said, this brings us to today and the birth of the Preterm Birth Prediction DREAM Challenge using microbiome data. We brought together vaginal microbiome data from 9 different studies that focused on pregnancy, preterm birth, and the vaginal microbiome to comprise our training data. The various data sets differed in the number of participants, samples, and data platforms as well. And many of these data sets can be found on the March of Dimes Preterm Birth Research Data Repository. But ultimately, the training data that was given to participants was used to accomplish 2 things. 1, predict preterm birth, which is defined as 37 weeks gestation, and 2, predict early preterm birth, which is defined as less than 32 weeks gestation. And so when it comes to the data, the time and frequency of the collection varied by study. Shown here on the left are the time points at which different samples were from different pregnancies were collected and broken down by study, as well as organized by term and preterm pregnancies. Studies also differed in the locations, as shown in the bottom right, and were mostly cohort studies rather than case control studies. Another major difference is that studies used 16S or shotgun sequencing. They had different variable region primers and used different sequencing platforms, Altogether, this provided both an analytic challenge as well as an opportunity. So some of the challenges of the challenge. Some of the initial problems that we encountered with the microbiome data is really rooted in the way microbiome studies are generally done. And the best way to illustrate this problem is I'd like you all to imagine sending a group of kids with cameras to a zoo and asking all of the children to take pictures of the animals that they saw. And so what you have here is a set of images that we've gotten back from the kids. And I'll ask you, what do you think are the different images or different animals that they saw at the zoo that day? And you can kind of guess because you know what animals and different animals look like, but the computational tools that analyze microbiome data don't. And as you can kind of glean from these images, all of the kids saw cats. And so you have different bits and pieces of the cat, but really unless you know what a cat looks like or have a good sense of what different cats would look like, it's pretty tricky to figure that out. And even if you do know what a cat looks like, it's hard to tell if they saw the same cat or if they saw different cats. And now at this point, you're just kind of using subtle hints, like the color of the cat, or maybe different patterns and stripes on the cat themselves, and so on. And really, it's not that different if you're looking at microbiome data, where you kind of take these raw pictures, sequences, and they kind of come back and tend to cluster more by the technique that was used to make them rather than the underlying biological differences. So in this figure is an ordination plot where each dot represents a sample and they're colored by the study from which the specimen came from. And so right here on the left, you see that different samples kind of cluster together based on the study. So teal with teal, blue and blue, red and red. And some of our validation studies from Wayne State University and Stanford, the ones that are outlined in black, kind of overlap with that green study as well. But altogether, it's a little bit of a mess. But what we've done is we've developed a novel technique called phylogenetic harmonization that can successfully bring this all together. And a slight tangent, but I just wanted to say thank you again to our colleagues at Wayne State University and Stanford for supplying us with unpublished vaginal microbiome data to use in this challenge. And so now with this phylogenetic harmonization technique, you have these bits and pieces of the cat and are able to bring them back together again. And so what you see on the right is after harmonization, all of the studies are really nicely integrated with each other. And the tool that was used in this phylogenetic harmonization approach is called Microbiome, which is short for the maximum likelihood amplicon pipeline created by Dr. Jonathan Golub. And as much as I'd love to go into great detail about the tool, it would be a little bit out of scope, but feel free to read the preprint and check it out at GitHub as well. And really, Microbiome was a great tool that helped us solve some of those initial problems that we saw in creating this challenge, but this wasn't our only problem. And so I'll pass it back to Tomiko.
Great. Thank you, Alennie. As Alennie mentioned, the problem of bringing the data sets together was something that we were able to use Microbiome to resolve, but there were many other challenges that we faced. Another one was, for example, with the features in the data sets and how, in a way, this contributed to failed submissions by our challenge participants, which people may know if they were involved in it. And actually, just out of curiosity, a show of hands, how many people here took part in our Microbiome Dream Challenge for preterm birth prediction? Okay, a few hands in the audience, great. So as you may recall, there were several features that we provided in the challenge data that you were able to work with, including phylotype, taxa, diversity, demographics, community state types, and more. and nearly all these features were generated by Maliampi. Having all these features was a good thing, we think, for our challenge, but it also presented a challenge. As individuals here may know, there's a lot of heterogeneity in the microbiome across time within any individual. As this slide illustrates, it shows the vaginal microbiome and how it changes over time in term pregnancy, preterm pregnancy, and early preterm pregnancy. Furthermore, between individuals, only about a third of our microbiome may be similar, meaning there's a lot of variation in microbiome across individuals. This brings us to what we saw during the challenge, is what some of the people here may have seen when they were participating, with Docker submissions failing, which was a real head scratcher for those of us who were organizing the challenge. But when we considered the heterogeneity of the microbiome across individuals, the biological sparseness of the data, it made sense why this was happening. The same features were not represented in the training and validation data sets. So what the participants saw, features-wise, may have been slightly different from what we had for the validation data. It's very simply illustrated in the top two tables here on this slide. Basically, there's so much underlying biological variability that some of the features weren't represented in both data sets. So what we needed to do, the challenge organizers, that is, we needed to make sure that the same features were represented across both data sets. And so that's very simply illustrated in the table down here below. We basically needed to create some additional columns, and some of those just had 0 values, but we needed to make sure that the same features were in both training and validation data sets. And as challenge participants may remember, there was an updated version of the training data set that we released a few weeks before the end of the challenge, and this was for that reason. And once the DREAM challenge was over, another challenge we faced was with validating models that our participants had submitted. For both task 1 and task 2, the DREAM challenge organizers, we had decided to use the area under the receiver operator curve to rank participants' models. And the two sub-challenges that we had were the prediction of preterm birth, which is the birth prior to 37 weeks of gestation, and the validation data set that we used for that consisted of samples collected no later than 32 weeks of gestation. For task 2, or sub-challenge 2, the prediction of early preterm birth, which is birth that takes place before 32 weeks of gestation, the validation data set included samples that were collected no later than 28 weeks of gestation. And to clarify, we chose to omit samples from the validation data sets that were a few weeks before the threshold for preterm birth or early preterm birth in order to minimize any potential for data leakage. When validating the persistence models, one issue we saw was that for binary predictions for task 2, for example, the early preterm birth prediction, some submissions had all zeros or just predictions of term birth, which resulted in some models giving a sensitivity of 0, meaning that the model was not picking up any early preterm birth cases. There were some concerns that the area under the receiver operator curve could result in good performance metrics simply because of the imbalanced nature of the validation data set, which consisted of 13 out of over 140 that were early preterm for task 2. So for our DREAM challenge, we wanted to be certain of participants' rankings. And so we took various strategies in order to validate participants' models. And these included bootstrapping, inverting probabilities, undersampling, as well as oversampling. And I just want to give kudos to Alice Tang, Jonathan Golub, Verena Chung, and Jake Albrecht for all their magnificent work in validating the models. On the top left, you can see the AU ROCs for a sub-challenge 1, which is the preterm birth prediction. And on the right, you see the AU ROCs when using a validation data set with inverted labels. That is, where term cases were labeled as preterm and vice versa, where preterm cases were labeled as term. And here we see that the AU ROCs, the rankings, particularly of the top teams, remains consistent. And actually, probably because of the really tiny font, you can't read the names, but that's okay. I'll show you who the winners are later. Down below, you see for the bootstrapping, what the AU ROCs were. And we bootstrapped 1,000 times, 100, we sampled 100 from 140 or so from the validation data set. And these are the box plots showing the ranges of the AU ROCs. And as again, you can see, if you can read the tiny font, that the top ranked teams remain the same. And here are the AU ROCs for the undersampling and the oversampling of the validation data sets. We have the bootstrapped AU ROCs for the undersampling on the left, oversampling on the right, and again, the top ranked teams remain consistent. This is the validation work for the second sub-challenge, which is the early preterm birth prediction. On the left are the AU ROCs from the sub-challenge that the participants saw. On the right are the AU ROCs from inverting the probabilities, and down below for the bootstrapping. And again, we see that the rankings, particularly for the top teams, remain consistent. And again, these are the AU ROCs for the undersampling and the oversampling from the validation data set. And we see the same consistency in the rankings. So after seeing that the top ranked teams were consistent after extensive cross-validation work, we felt comfortable and confident in announcing who our winners are, which bring us to the results of our Dream Challenge. By the time our Dream Challenge ended, we had 318 individuals and teams register for our challenge, and we received 148 submissions for sub-challenge 1 and 121 submissions for sub-challenge 2. And here are the winners of our preterm birth prediction Dream Challenge. For task 1, the University of Wisconsin-Madison team and the AI for Knowledge lab are our first and second place winners. Their models were able to achieve AU ROCs of about 0.69 and 0.64 respectively, which was better than our baseline models. For task 2, the Techman lab and the KBJ team were our first and second place winners, and their models were able to achieve AU ROCs of about 0.87 and 0.84 respectively, again, better than our baseline models. One thing that we saw in these teams and other teams, top-ranking teams submitted models, was that many used ensemble methods for their predictions. And we are very fortunate because we have members from these winning teams here today who will be talking with you about their work a little bit later during this session. So what's next for those of us who are organizing this Dream Challenge? Well, our immediate next step is to work with challenge participants and to share our findings from this Dream Challenge to identify pregnancies at risk for preterm birth. We aim to dive deeper into the findings and learn how it is that the microbiome may increase or decrease the risk of preterm birth. And ultimately, it is our hope to reduce the risk of preterm birth by possibly changing the microbiome. And with that, there are many people that we would like to acknowledge, including members of the Sirota Lab, Marina Sirota, Alice Tang, Alennie Roldan, Jackie Roger, Jean Costello, Brian Lee, Edie Costi, Gaia Androletti, and many, many others who help provide great insight and help into this challenge. The UCSF microbiome colleagues, including Susan Lynch, Connie Ha, and others, who helped us with processing some vaginal microbiome samples and the data we were able to use as part of our validation data set. Our Periton colleagues, who help us with developing and maintaining our Marchadimes preterm birth research data repository, and members from Stanford, including David Stevenson, Nima Agepor, Ron Wong, who generously shared vaginal microbiome data sets that we were able to process and use that data as part of our unpublished validation data set. Of course, members of our DREAM team, including Jonathan Golub, Jim Costello, Adi Tarka from Wayne State, he shared unpublished vaginal microbiome data that we were able to use for our validation data set. Verena Chung, Caitlin Flynn, Jake Albrecht, and many, many others from the DREAM team, without whose help organizing this Dream Challenge would not have been possible. And of course, the Marchadimes, Emery Selly, Jonathan Cherry, Courtney Dower, and many, many others Without their very generous funding and support, we would not have been able to bring this challenge together. And I just wanted to mention that the March of Dimes sponsors 6 prematurity research centers that each has different themes with respect to their research work in preterm birth. And our March of Dimes Prematurity Research Center at UCSF, led by Marina Sirota, is involved with data sharing, our data repository work, crowdsourcing approaches, as well as computational drug repositioning, and leveraging the electronic medical record data to study pregnancy outcomes. Our preterm birth research data repository, which includes microbiome data, in addition to the data that we used, a lot of it came from the March of Dimes data repository. We also have GWAS data, proteomics, CyTOF, and a lot more. I invite you to visit pretermbirthdb.org for more information, as well as to get data for your for any work that involves preterm birth research. And this is a picture of people from our lab at an outing out in San Francisco on a sunny day, which is not common in the summer. So anyway, thank you very much for your time and attention, and we're happy to take questions. Sure.
So to describe, so we talked about microbiome data. Can you describe a little bit more about what that is? So obviously, you know, bacteria something, but can you give us a sense of the variety and you know, how deep is that sample? What's the technologies that we use to sample that data?
Yeah, so the vaginal microbiome, well, that's what we use in this particular challenge, and that's collected from, for this challenge, collected from patients who were pregnant all throughout different times, usually with a swab, and those can be frozen or otherwise preserved and then sequenced. And so what we're sequencing is the genetic material from these organisms, so from the bacteria, from the viruses, from the fungi, all these different organisms that live within us. And there are various techniques. 16S is one method. There's also whole genome sequencing, where you sequence the entire genome of these organisms. And the tricky thing with the 16S method that we were talking about is that they can use different regions. They're called sequence variants. They can use different regions for the sequencing. So that's why, like that analogy that Alennie was showing you earlier, you end up getting different pictures of these organisms that you're trying to bring together when you're doing a meta-analysis. And that's the tough thing. But when you have a tool like Maliampi that's able to homogenize these data by using what's called a phylogenetic reference tree. So it's like having the whole picture of the animal, and you're able to take all these bits and kind of hang those little bits onto these big pictures, then you have a sense of what it is represented in your data.